Jean Sedibir in 1782 proved that plants have observed, they absorb carbon dioxide and they release oxygen in the presence of light. He also showed that the rate of oxygen evolution depends upon the rate of carbon dioxide that was consumed. Nicolas de Saussure, 1804, he showed that the importance of water. He showed the importance of water in the process of photosynthesis. He further showed that the amount of carbon dioxide that's absorbed is equal to the amount of oxygen that is released. Julius Robert Mayer, 1845, proposed that light has radiant energy and this radiant energy is being converted to chemical energy by plants, which serves to maintain the life of the plants and also the animals. Julius von Sachs in 1862 demonstrated that the first visible product of photosynthesis is starch. He also showed that chlorophyll is confined to just the chloroplast cells. Thus, Julius von Sachs laid the foundation of the modern concept of photosynthesis. An interesting experiment done by T.W. Engelmann, 1843, from 1843 to 1909 he lived, by using a prism. He showed the experiment that it splits light into spectral components and then it is illuminated a green algae, Cladophora. The name of the algae was Cladophora, and he placed in a suspension of aerobic bacteria. So you see, this is the prism, this is the light, and you could see that how the prism is splitting the light, the spectral components. So you see the Cladophora cells, chloroplast, which practically fill each cell, and the motile bacteria over here. And you see the prism when it splits the white light. In Engelmann's experiment, the bacteria were used to deduct the So basically they detected the site of oxygen evolution, from where oxygen evolved. So this is the aerobic bacteria. He observed that the bacteria accumulated mainly in the region of the blue and the red light of the split spectrum, and at first action spectrum of photosynthesis was thus described. The first action spectrum of the photosynthesis resembles roughly the absorption spectra of chlorophyll A and chlorophyll B. Remember this. Again, in 1930, C.B. van Niel proved that the discovery of Nicolas de Saussure was wrong. C.B. van Niel showed that sulfur bacteria used the hydrogen sulfide in the place of water and carbon dioxide to synthesize carbohydrates as follows. And this was the reaction that he gave. That is, carbon dioxide combines with sulfur dioxide, gives you glucose, water and sulfur. This led Van Niel to postulation that in green plants, water, that's H2O, is utilized in the place of H2S and water, that's H2S and oxygen is evolved in the place of sulfur. So now just replace the whole thing. You see here 6 CO2 carbon dioxide plus 12 H2O water when combines in the presence of sunlight, it gives you C6H12O6, that's glucose, plus 6 H2O, that's water, plus 6 O2, that's oxygen. So from this, our photosynthesis reaction came into existence. The discovery of Van Niel was confirmed by Rubin and Kamen in 1941 using chlorella, another green algae. Rubin and Kamen used water that had a heavy isotope of oxygen, that is O raised to 18, and found that oxygen released in the process of photosynthesis was the same one, that is O18 type, that was the heavy oxygen. On the contrary, one Carbon dioxide 18 was used. The released oxygen was of the normal type, that is oxygen O raised to 16. That was the type. So from this experiment, it can be concluded that oxygen released by the green plant comes from splitting of water and not from carbon dioxide. Huber, Michael and Diessenhofer in 1985 crystallized the photosynthetic reaction center from the purple photosynthetic bacteria, that was Rhodopseudomonas viridis. They analyzed its structure using the X-ray diffraction technique. In 1988, they were awarded the Nobel Prize in Chemistry for this very work and this contribution.